I'm David Sadava. I'm a professor at the Claremont Colleges. I'm also a scientist. I do biological research in cancer chemotherapy, so I know a fair amount about cancer. In fact, I helped on the video series that you saw, if you did, last week on PBS. Want to talk a little bit about biology education, and especially a couple of reports that came out about biology education over the last dozen or so years. The first one in the upper left is called Biology 2010. Obviously, that came out before then. It was the early 2000s. The second is called Vision and Change in Biology Education. That came from the American Association of Advancement of Science. The third one that you may not know about is the MCAT, the Medical College Admission Test that all pre-medical students must take for admission to medical school. And that is a new test this year, and the report came out about a year and a half ago. And it also examined the teaching of biology, general biology, at the university level. So these first three reports, the Bio 210, Vision and Change, and MCAT, dealt with biology education at the undergraduate university level. And then, of course, you're very familiar with the AP biology report. All of these reports, to me, highlighted the same issues. And here they are. First, biology teaching these days, according to the reports, had too many facts to memorize. We were just cramming too much information into our students. Second, the reports all said there's not enough emphasis on how biologists really think about biological problems. And that could go to applied biologists as well, farmers or physicians. Third, there was little consideration of what are called biological systems. In recent years, the recent meaning the last 2 decades or so, a lot of biologists in fields related to biochemistry, for example, ecology, have used an analysis called systems analysis, where you look at interacting parts and you find that the whole is greater than the sum of the parts. And systems analysis comes from engineering. Fourth, very interesting to me, there were complaints that there was inadequate consideration of the applications of biology to society. It seemed that we were teaching students a whole lot of facts about DNA or about phyla or about organisms, and not about how they fit into our society, from ecology issues to medical issues to agricultural issues, food production, etc. Well, here are a couple of highlights from the biology education at the university level reports that came out that I mentioned. There were 3 of them: Biology 2010, you see in the upper left, Vision and Change, and the MCAT. So first, Biology 2010. If I could highlight 2 things there, first of all, it was the idea of biological systems, that biology education really was ignoring systems in biology and how, as I mentioned, the whole sometimes is greater than the sum of the parts. The second thing that Biology 2010 really pushed was databases and computer modeling. This especially was genomics. This report was written by a bunch of people who were very much in thrall to the recently revealed human genome and other genomes as well. And there's a huge amounts of data, and they even got the idea that all biology students should take computer science and should be able to do the modeling on their own. So this was a very math, computer science-oriented report, as well as a systems biology report. Now the middle one, Vision and Change. There were some of the same people in the Vision and Change as there were in Biology 2010, and some different, again, at the university level. Here, they really took biology to task for not emphasizing core concepts of biology, and they listed several core concepts. You can imagine one of them was evolution, another might be energy flow. Second, core competencies. They wanted to really list what are the competencies we want our students to have. And this is a current thrust in undergraduate education where we're trying to get students to master certain competencies before they graduate, or in this case, after they've taken an introductory course. So core concepts, core competencies. You'll see something like that in a moment. The third report I'm referring to is the one in the lower left, which is the MCAT, the Medical College Admission Test. And you might say, well, who cares? That's just a test for pre-medical students. Well, a lot of students go into university wanting to go to medicine. It seems that sometimes that my students don't know that there's any other way for a biologist to act. Some of them will even tell me that I should have done this, become a physician rather than a scientist. The MCAT obviously is very human-oriented, so they revise their biology topics to be much more human-oriented and problem-solving. And secondly, and very importantly, they now require advanced biochemistry knowledge and psychology and sociology concepts as well. And this is going to have enormous impact on undergraduate education at many, many universities, because after the introductory course, students will be taking a course in biochemistry, a course in psychology, and a course in sociology if they're intending to go into medicine. Or it's going to, you'll see, the other health science professions will follow as well. This might not be a bad thing, having them knowing psychology and sociology. What's the result of all this? Well, I have the result in the bottom line. There are about 6,500 2- and 4-year colleges and universities, and there are 6,500 different biology courses. We all read these reports, and we all said, gosh, that's really interesting. We better cover these topics, for example, and almost everyone covered the topics. But everyone has their own flavor, and all the courses are different. You people who are in AP have done a lot better job. So here's the AP report and the AP biology report. And the AP biology report, as you know, came from the ground up. It was a representative group of teachers, of university professors, of scientists, of people in schools of education, etc. And they came up with 4 big ideas and 6 science practices. Oh, is it looking familiar to you from the previous report on vision and change, core concepts, core competencies? Well, here they're called big ideas and science practices, and they are rather similar to one another. The result of AP is very different than the result of 6,500 biology courses, and all different. There are approximately 10,000 high schools, the last I heard, who are teaching AP biology, and there's only one course. And that one course you are all involved with probably is following the AP biology's 4 big ideas and 6 science practices. Now, several of us were sitting around one day with a publisher, and we were thinking about all these reports that had come out and all the talk there was about teaching introductory biology. And we decided that there was a need for a new book and a new approach at the textbook level and associated materials with the textbook for teaching of biology. And this might be consonant with the principles and concepts that had come out in these 4 reports. So what did we do? Well, we read the reports, or we read drafts of the reports. For example, our meeting of our team came when the biology education and the AP and the biology education vision and change and the biology education MCAT were under preparation, but we had drafts of those reports. So we decided to invite to a retreat 20 people, ranging from high school teachers to people involved in the design of AP. We had Bill Wood, who is a friend of mine, who was the chair of the AP committee for AP biology. We had people from all over North America, again, ranging from high school to college to large university to small colleges. And we sat around and talked about biology education and some of the new ideas that were percolating and asked them, what do you envision in a book? And a couple of years ago, out came Principles of Life, the first book that was built from the ground up by a representative group, some from the AP committee. This book was concepts rather than facts-oriented, just what the reports had been asking for. It had an emphasis on, and has an emphasis on problem-solving and experiments rather than just packing a lot of information in. And this book, again, it was the first book of any in mainline textbooks that really addressed the concerns of the reports in biology education. And we were thrilled. It was very well received, and it was especially widely adopted by AP biology teachers. Many AP biology teachers are using our book. We learned from the first edition, and now we have a second edition, Principles of Life, second edition. This book, by the way, is published by, co-published. It's published by Bedford, Freeman, and Worth, which is the organization that is sponsoring this webinar, and also by a boutique publisher called Sinauer Associates, who works with our author team on the actual content and design of the book. Well, let's look at AP Biology in a little more detail. Here are the big ideas of AP Biology that I'm sure you're familiar with, and I've paraphrased them here to get them into one sentence or one phrase. First of all, evolution drives unity and diversity of life. The second big idea, biological systems use, there's that word again, use free energy to build and molecular building blocks. Third, living systems store, retrieve, transmit, and respond to information. Fourth, biological systems interact and possess complex properties. So these are the four big ideas that are in the AP Biology manual. There are 6 science practices for AP Biology that you may be familiar with. First, using representations and models to explain biology. Second, using mathematics. Third, engaging in scientific questioning. Fourth, planning and implementing data collection strategies. Fifth, performing data analysis and evaluation of evidence. And finally, working with scientific explanations and theories. And the hope is, in combination with a book and a classroom and a laboratory, and we are familiar with the laboratories that you use in AP Biology, all the combination will give a student the science practices and, as I mentioned before, the big ideas in biology. And then they can go on from AP to advanced courses at the university level or even at the high school level. Well, what I decided to do and help you with our book is to show you how our textbook, which is Principles of Life, in this case the second edition, relates to the science, the big ideas, and the science practices of AP Biology. And at random, and literally at random, I just opened up the book and I said, well, let's take one chapter. And the chapter I've chosen is our chapter 19, which happens to be a diversity chapter. I'm not a diversity biologist per se. I'm a molecular biologist. But I chose this chapter just to show you all the ways that our book applies to the 4 big ideas and the 6 science practices. So let's take a look at it from this particular chapter. Some of you may have the book. If you have it, you can open up at chapter 19. If you don't, that's okay too. I'm just going to give you page references, but I'm trying to make a point, and my point is we could open any chapter of our book and I could show you the same thing. So let's look at the first big idea, obviously evolution. Evolution permeates our book, and we use a lot of tree analysis. This shows you a tree of life for living things. We have the 3 major branches: bacteria, archaea at the bottom, and eukarya in the middle in blue. And we have different gene flow as little arrows here. But the idea, this is on page 379 of the book, is this is relating organisms through evolution and natural selection. And that, again, permeates, and it would permeate any biology textbook. It certainly permeates our textbook as well. We have an appendix in our book, the only book that has this, on the tree of life, that essentially is a searchable appendix online where you can search any class of organism and get all the information on that organism on the tree of life, relating one organism to another. So, big idea number 1, evolution. Big idea number 2, energy. Giving you 2 examples. We decide, and we really enjoy doing this, we open up every chapter of our textbook with an opening story. Now, there's a risk here because the students are going to say, it's not on the test, I'm not going to read it. But we found that most students are really engaged by our opening story. This is a night view of the Earth, and this is a glowing piece of the sea, region of the sea, off the coast of Africa, which a fishing boat saw, and the fishing boat saw the sea essentially glowing, which is incredible. It's a mat of Vibrio, which is a bioluminescent organism. So we talk about the small organism and how it uses light to produce energy, and that gives it the energy flow. Here's another example of energy we're talking about here in this chapter in 385, talking about cyanobacteria being the first photosynthesizers, and of course photosynthesis is a topic that we cover in great detail in other chapters. So, big idea number 2, energy. In this chapter on viruses, and bacteria and archaea. Third idea, big idea number 3, information flow. Obviously, we're going to deal here with DNA and its uses. In this particular chapter, we look at gene flow. So we're trying to show in this figure, for example, that just classifying organisms on their gene sequence might be misleading because genes have hopped or moved from one organism to another in lateral gene transfer. So we're inferring phylogenetic relationships, and we've got to be careful in doing it if we have organisms where genes have moved from one organism to another, and we give an example here. Again, using tree analysis, but the information content of tree analysis. The fourth big idea, interacting systems. This is a biofilm. A biofilm, in this case, is a lot of prokaryotes who are free-living, and they signal molecules to each other, and they form this film. This is a photograph on the right, electron micrograph of dental plaque. I just came from the dentist this morning where I got my cleaning, and I was thinking of this photograph as it was done. And this is a biofilm showing how the organisms relate to one another to form more or less a superorganism that has properties that are greater than the sum of the parts. Again, in this particular chapter, we're showing big idea number 4, which is the interaction of systems. I mentioned there are 6 science practices. Here's science practice number 1. Representations and models. I'll give you 2 examples. First one is on page 389 of the book. And this is the familiar model to all of you of the bilayer membrane, and in this case we're looking at different compositions of fatty acids, so we're representing it in the model by straight or kind of fairly rigid lines versus wavy lines, right? It's really a model. The membrane doesn't actually look like this. Some of my students actually think that there's little blue balls. It's not. This is a model representing reality. Of course, doing analysis of tree analysis is another way of modeling relationships between organisms. So representations and models permeate our book, and I'm just showing it in this particular chapter. Science practice number 2, using mathematics. We really were the first book that really pushed using mathematics in the book. So for example, here is an instance where in an experiment we're asking students to plot some data. So this is temperature versus generation time, and we're using, what we're interested in is, of course, the optimum and temperatures that organisms survive and lethal temperatures where they're killed. And this is generation time in hours, and we ask students to make a graph, etc. Applying the concept, we have a very interesting idea that we pioneered in our first edition of applying all the concepts that we have in real-world examples. So this is viruses evolving over time, and this is the genome size on the y-axis, and on the x-axis the number of protein-coding genes. And we show that free-living bacteria have many more genes and a larger genome than bacteria that are symbionts or parasitic bacteria, and there are many implications of that. And then we ask the student again to analyze this graph. So using mathematics is very important. We also use statistics in our book. We are the first book to have a statistics appendix, and it's not just plugging in numbers. All of you know that there are websites the students can go to. The one we use is Vassar Stats, where students can go and just plug in numbers and get standard deviations and standard errors, etc. if you're teaching that. We have a statistics appendix that shows not deriving all of these equations, but where they come from, why you would use a test in a different situation, and then we actually ask the students how you would use statistics to validate or invalidate a conclusion. So that's science practice number 2, using mathematics in this particular chapter. And again, the chapter is on bacteria, archaea, and viruses. Science practice number 3, scientific investigations. Well, we set off our figures of investigations. We go through hypothesis, method, results, and conclusion. And this is really the way scientists do it. You will see almost no papers in the literature that I am familiar with using null hypotheses, etc. We don't do that formally. We are generally developing a hypothesis and then testing it, and then seeing whether the test results validate the hypothesis. You'll note here we have little balloon captions to show what happens. This is an experiment where we're seeing whether organisms will survive at temperatures above 120 degrees, which is quite a high temperature, and they identified a prokaryote from water samples in a hydrothermal vent. And this is an actual experiment that was in the literature, and then we have data from the literature here. So investigation, hypothesis, method, results, conclusion. And then we ask actually students to discuss that later on. So students see what scientific investigations are. Again, in this particular chapter, but I could show you this in any chapter. Data collection. Well, in this chapter, we're doing data collection, and we show the microbiome, which you may be aware of. It's the collection of microbes that inhabit our digestive, reproductive, and respiratory tracts in the human or in another organism, large organism. And here there's a collection, and we're looking at ones that were sampled and organisms that were defined by their genome sequence. And we're sampling, right? We're collecting data, and we're essentially catalyzing the number of different organisms, that's on the y-axis, from different parts of the body, from the skin, from the reproductive tract, from the respiratory tract, etc. So data collection. The fifth science practice, evaluation of evidence, and this is where we're very strong. In every experiment that we report, as well as some of our boxes of analyzing information called Applied to Concept, we have real data. These are real data from the original scientific article. You know, you got to be careful when you choose data for presenting to high school students or to undergraduates. You've got to choose data that's going to lead them somewhere that's not overly interpretable, and that's also not too complicated, because they see something of 15 decimal points. It may be quite a chore for them to look at. So here's that table I just showed you, and we have this section called Analyze the Data. So here's the temperature versus generation time, and we ask the students to make a graph, as I mentioned before, and then we say, what can you conclude? What temperature appears to be closest to the optimum temperature for this strain? And then we say in part C, notice no growth occurred at 130, but the cells were not killed. And then we ask the students to propose an experiment. How would you demonstrate that these cells were still alive? So we are not only looking at the evidence, we're evaluating the evidence, and we're also proposing other experiments that you could do based on the evaluation of that evidence. That's science practice number 5, and once again, I could show this for any chapter of the book. Finally, scientific theories. How do you test what AP asks us to do is to teach our students how you propose and then test the scientific theory. Well, we had a really good one in this chapter. It's Marshall and Warren from Australia satisfying Koch's postulates. Koch's postulates were developed in the 1800s by the great microbiologist Robert Koch, and he was trying to prove, that is, to give estimates, to give evidence that a microbe causes a disease. Mere association doesn't do it. You've got to do a series of experiments. In this case, Marshall and Warren did the experiment on themselves. They wanted to prove that an organism, a bacterium, actually causes stomach ulcers, and that went against everyone's thought at the time. And so they essentially did Koch's postulates on themselves. They essentially did the test. They took the microorganism in the disease, that it was in the people with ulcers. They cultured the microorganism from a person with ulcers. Then they essentially induced the disease in themselves by swallowing the microorganism, and they induced some inflammation that was pre-stomach ulcer. Then they recovered the bacteria from the infected individual, and then they repeated it. So, and they used antibiotics, of course, to eliminate the bacteria, and that's the standard treatment now for a lot of ulcers, stomach ulcers. Marshall, by the way, won the Nobel Prize for this particular experiment. But it's a very, very clear and nice example of using a scientific theory, again, in this particular chapter. So, in AP Biology, we have taken all of the big ideas, all the science practices, and shown, in this chapter, for example, again at random, that our textbook essentially will cover all of those. To help you with the book, we're preparing a new teacher's edition. It's just about done. I believe it'll be done in the summer. completed in the summer, and you're going to receive, I think, a sample chapter. This is written by and for AP Biology teachers. It's in a wraparound format, and the guides, again, it's got the big ideas, science practices, and it's got a terrific number of exam and teaching tips. It's got mini labs. It's got tips for the AP exam. Again, in the margins, in a wraparound format. It's got teaching tips. One thing that it has that I find rather interesting is that essentially it tells you what's on the AP exam and what's not. So our book has a lot more in it, as any book does, than is covered in the basic big ideas and science practices in AP Biology. And so it actually, this guide actually points those out and says, you know, this is interesting, but it may not be on the AP. It's not on the AP guide. It's not on the AP test. So maybe you'd like to be light on this and not emphasize it. I'm glad that's what's said rather than skip it, because as an author, that hurts my feelings. It's a terrific guide. I'm just amazed at what's been done. I've been consulted on it, but I haven't written it. It's been written by AP Biology teachers for AP Biology teachers. And again, it's going to be a very, very useful thing for those who use the book. Well, I'm in reverse order. I want to introduce us first. I usually introduce the speakers and the authors first. I'm introducing us last. I'm in the lower left. I'm David Sadava. I'm a cell biologist, molecular biologist. I held my doctorate from the University of California at San Diego. I teach courses in general biology for majors and non-majors. I teach cell biology, biochemistry. I taught one of the first cancer biology courses in the country, starting about over 20 years ago, at an advanced level, again, showing students how you can use biology principles to look at the cancer problem. I do research in the laboratory where I just was. I do research on cancer chemotherapy for lung cancer, which you may know is a very disastrous cancer, which is the leading cause of cancer death. On the upper left is our senior author, David Hillis. David is an evolutionary biologist at the University of Texas at Austin. He was one of the authors of Biology 2010, as well as on the committees involved with Vision and Change. On the upper right is Mary Price, University of California, Riverside. She's a community aquatic ecologist, does research at Rocky Mountain Laboratory in Colorado, and has taught general biology many, many times, as have all of us, by the way. And the lower right is Dick Hill. Dick is a animal physiologist, but is very interested in comparative physiology rather than human physiology. And he keeps us all on our toes so that we don't only use human examples in physiology. We are very much aware of every organism, including plants, and I was originally trained as a plant biologist. So that's our team. That's our book.